fellow warriors. Here we are with news from the website, prostatewarriors.com.
Yeah, and today we really have some incredible stuff to get into.
We really do, because our mission today is to take all this dense, heavy medical data on the latest breakthroughs and actually translate it into something actionable for you.
Right. We want to look at how the entire battlefield of prostate cancer is shifting right now.
Exactly. I mean, we're going to start by looking at the latest active clinical trials, and then we're moving into some really cutting-edge preclinical strategies.
Yeah, the lab work right now is fascinating.
It is. And we'll wrap up with a totally surprising discovery involving, of all things, a super famous household drug.
I think the last one is going to genuinely shock a lot of people listening.
For sure. But let's start right on the front lines. There's a major regulatory shift happening with how we treat metastatic hormone-sensitive prostate cancer.
Right, the MHSPC stage.
Yeah. So we're looking at the Phase 3 PSM Edition trial. And the big news here is that 177 Lu PSMA 617.
Which is Pluvicto.
Right, Pluvicto. It's moving up to be a first-line treatment combined with standard androgen deprivation therapy.
Which is a huge structural shift in how we approach this.
It really is. I mean, they saw a 33% reduction in the risk of progression or death.
Yeah, that 33% is a massive number in oncology.
But, my first thought when I read this was a military analogy. It feels like we're taking our absolute heaviest artillery and just moving it straight to the front lines instead of, keeping it in reserve.
That's exactly what's happening.
But isn't there a risk there, like playing your best card way too early?
Well, yeah, I mean, that's been the debate for years, right?
Mm-hmm.
Historically, you always hold the big guns back because you just assume the cancer is going to mutate eventually.
Right, you want to save something for later.
Exactly. But this trial really challenges that old wisdom.
Yeah.
And to understand why hitting it early actually works better, you have to look at tumor heterogeneity.
Basically meaning how diverse the cancer cells are?
Yeah, exactly. Because early on, in that hormone-sensitive stage, the cancer cells are mostly uniform. They all look and act relatively the same.
Okay.
But as the disease progresses and you start throwing different treatments at it, the cells that survive, they mutate.
They adapt.
Right. They become incredibly diverse. So by the time a patient hits the castration-resistant phase, you aren't just fighting one type of cancer cell anymore. You're fighting, like, dozens of subclones.
Wow. And they all have different defenses.
Exactly. So by bringing 177 Lu PSMA 617 early, you're hitting them when they're vulnerable and uniform. You can wipe out a huge portion of them before they even have the time to build up those heavy mutations.
Oh, I see. So you're basically stopping them from digging those really deep, complex trenches in the first place.
Yes, you're literally altering the evolutionary trajectory of the tumor.
Because the 177 Lu PSMA 617 hunts down the PSMA positive cells and just drops the radiation right on them. So you dramatically delay the onset of that castration-resistant phase.
That's the goal, yeah.
But, okay, what happens when the cancer does figure out a workaround? Because some patients listening are already in that advanced castration-resistant phase where the standard options are pretty much exhausted.
Right, and that's where we need totally new weapons.
Which brings us to the secure clinical trial. So this is for MCRPC, and we're looking at a completely different radioligand here. 67 QSAR bis PSMA.
Yeah, that's a really exciting one.
So the trial gave a cohort of 19 participants an 8 gigabecquerel dose, and the results are just wild. 7 out of the 19 patients actually achieved a complete response or undetectable disease after a median of just 2.8 cycles.
The depth of that response is what's really turning heads.
Yeah, I mean, 63% had a PSA drop of at least 50%, and over a quarter of them hit a PSA 90.
90% reduction.
Right. So walk me through this, because the actual structure of this drug sounds completely different from Pluvicto.
It is. The structure is what makes it so aggressive. So it uses copper-67 as the radioactive payload, but it's the delivery mechanism that's key. It uses a bivalent or a bis structure.
When I read bivalent, I just pictured like 2 hands grabbing onto the target instead of one, like a grappling hook with 2 prongs.
That is basically the perfect way to visualize it. And that dual binding setup is absolutely critical.
Why is that?
Well, the tumor microenvironment is incredibly chaotic. It's hostile. So a standard single binding ligand can sometimes get knocked loose or detach before the radiation fully deploys.
Oh, it falls off before it can finish the job.
Right. But by using that bivalent two-handed grab, the 67 QSAR This PSMA locks on with massive stability. It just holds on tight.
Ensuring the copper-67 actually hits the DNA.
Exactly. And copper-67 is a highly energetic beta emitter, so you want it anchored directly to the cancer cell to do maximum damage.
Okay, but I have to play devil's advocate and pause you there for a second.
Sure, go ahead.
I'm looking at the trial size, 19 patients.
Yeah?
I mean, from a data perspective, 19 is a tiny cohort. If a pharma company brought me a study of 19 people, I would be extremely skeptical. Are we jumping the gun getting excited about this?
Honestly, if this were an early-stage disease trial, I would 100% agree with your skepticism.
Right.
But context is everything here. These are patients with metastatic castration-resistant prostate cancer. They've already progressed past all the standard androgen receptor therapies.
So they're in a really tough spot.
Extremely tough. The disease is deeply entrenched. Achieving a complete response, where the disease is literally undetectable in this specific group, is just incredibly rare.
Wow. Okay, so even though 19 is a small sample, seeing 7 complete responses is huge.
It's a profound signal. It proves that this underlying mechanism, that two-handed bivalent grip with the copper-67, can actually break through the cancer's most heavily fortified defenses.
So it's basically a proof of concept in the harshest possible environment.
Exactly. It validates the engineering of the drug itself.
Okay, that makes a lot of sense. So radioligands drop bombs directly on the target. But what if we don't want to use radiation? What if we want to enlist the patient's own immune system?
Which is the holy grail of oncology, really.
Right. Well, we have news of FDA clearance for a new cellular immunotherapy study. This is the Bria-PROS plus therapy, and they just got cleared for a phase 1/2, a trial.
Yes, and the design of this therapy is fascinating.
They describe it as an investigational, personalized, off-the-shelf cellular immunotherapy using human leukocyte antigen matching, or HLA matching.
Which is quite a mouthful. But combining off-the-shelf with personalized is usually an oxymoron.
Right.
But it represents a massive leap in logistics, because building bespoke cellular therapies from scratch for every single patient takes a ton of time and resources.
And time is exactly what patients with advanced disease do not have.
Exactly.
You know, when I was reading about the HLA matching part, it reminded me of traveling internationally.
Oh, yeah. How so?
Like, when you travel with sensitive electronics, you have this really powerful piece of equipment, which would be the off-the-shelf therapy. But if you just jam it straight into a foreign wall socket, it shorts out.
Right.
You have to have the exact right adapter plug, and it feels like the HLA matching is that adapter plug. It makes sure the therapy interfaces perfectly with the patient's immune system without causing a massive rejection.
That's a great way to look at it. I'd actually take that analogy even further.
Okay, let's hear it.
If you use the wrong adapter overseas, not only does the equipment not work, but you trip the circuit breaker for the whole house.
Oh, wow. Yeah.
In the human body, if the HLA doesn't match, your immune system sees the therapy as a foreign invader. It actively destroys it before it can even touch the cancer.
Well, that's terrible.
But when you get the adapter plug right, Bria-PROS Plus can actually go to work. And what's really unique here is that it stimulates both the adaptive and the innate immunity.
Right. The preclinical data says it activates naive T cells, dendritic cells, and natural killer cells.
Yeah, all three.
Wait, why do we need all three, though? We always hear about T cells in the news. It seems like T cells are the only ones that matter in immunotherapy. Why isn't just sending in the T cells enough?
Because prostate tumors are master manipulators. They're notoriously good at creating an immunosuppressive microenvironment.
Meaning they shut the immune system down?
Exactly.
Yeah.
Imagine the tumor building a chemical wall around itself. And that wall emits signals that tell your immune cells to just power down and go to sleep.
So the T cells just get neutralized.
Right. The chemical wall puts them to sleep. So to breach the wall, you need a highly coordinated assault.
A multi-front attack.
Yes. The dendritic cells are like the scouts. They gather intelligence and show the rest of the immune system exactly what the cancer looks like.
Okay.
Then the natural killer cells are the brute force first responders. They don't need complex signaling. They just start attacking immediately.
Love that.
And then the T cells come in to provide the long-term specialized memory so the body keeps hunting the cancer over time.
Oh, I see. By activating all three at once, you just overwhelm the tumor's ability to suppress the response.
Exactly. You wake up the whole battalion at once.
Okay, so we've covered the heavy artillery of Pluvicto, the bivalent grappling hooks, and this multi-front immune assault. Now, let's step away from the active trials and look at the preclinical lab work.
This is where things get Really futuristic.
Seriously, this is where the next generation of targets is being mapped out, and the tools they're using are just unbelievable.
Yeah, we're seeing huge leaps in how researchers are hacking antibody-drug conjugates, or ADCs.
Right. So let's look at the JU Panorama Single Cell Atlas. The scale of this study is hard to even comprehend. They analyzed nearly 1 million individual cells from 213 patients.
Just a massive amount of data.
And through all that, they identified this target called B7H3, also known as CD276. And it turns out this target is highly stable across all states of prostate cancer.
HSPC, CRPC, neuroendocrine, all of it.
Right. But here's the piece of data that literally stopped me in my tracks. Unlike almost every other target, B7H3 is actually repressed by the androgen receptor.
Yeah, this is brilliant.
Wait, does this mean what I think it means? If standard hormone deprivation therapy shuts down the androgen receptor, does that mean our standard therapy actually makes the B7H3 target more visible?
Yes. You hit the nail right on the head. That's the pivotal finding.
Wow.
Most of the targets we use require the androgen receptor to be active. So when a patient goes on standard hormone therapy to starve the cancer, the androgen receptor shuts down. And unfortunately, a lot of our therapeutic targets just vanish from the surface of the cell.
So it's like we turn off the lights to starve the cancer, but by doing that, we accidentally blind our own targeting systems.
Exactly. But B7H3 works in reverse. By suppressing the androgen receptor with standard therapy, you actually remove the genetic brakes on B7H3.
So the target doesn't vanish, it lights up.
It lights up like a beacon. The researchers use computer science terms to describe this. They call it and/or gating logic.
Okay, explain that gating logic for me. I want to know how that translates into a real treatment strategy.
Think of it as a trap, a synergistic trap. You design a protocol combining a B7H3 targeted drug like MGC018 or DS7300A with standard androgen deprivation.
Okay.
You're creating a scenario where the cancer cell is doomed no matter what it does.
Right.
If it relies on the androgen receptor to survive, the standard hormone therapy kills it.
Right.
But if the cell tries to mutate and survive without the androgen receptor, it accidentally turns on B7H3.
Oh, wow. So it essentially paints a giant bullseye on its own back.
Exactly. And then the antibody-drug conjugate just locks onto that bullseye and destroys it. You're using the cancer's own survival mechanism against it.
You literally force the cancer to choose its own poison. That is incredibly elegant.
It really is.
And speaking of cutting off escape routes, UCLA researchers just found another way to hack these ADCs. They figured out that pairing DNA-damaging ADCs with a BCL-XL inhibitor vastly improves the effectiveness.
Yeah, this is a very clever approach.
Walk me through the mechanics here, because I know it ties into how cells naturally decide to, well, die.
Right. It's called apoptosis, programmed cell death. Normally, when an ADC drops a toxic payload that shatters a cell's DNA, the biological response is to initiate that death sequence.
It self-destructs.
Yeah. There's a gene called TP53 that acts like the supervisor. It sees the massive DNA damage and pulls the plug on the cell.
But cancer cells don't want to die.
Right, they're survivors. So they overexpress this protein called BCL-XL to block that death sequence.
So TP53 is yelling, the damage is too severe, shut it down. But the cancer cell uses BCL-XL to build a dam and block the signal.
Exactly. The BCL-XL patches the holes and keeps the zombie cancer cell alive, even though its DNA is wrecked.
So what did the UCLA researchers do?
They introduced a BCL-XL inhibitor alongside the ADC.
Oh. So the inhibitor just rips the patch right off the dam.
It completely disables it.
Mm.
By inhibiting BCL-XL, you take away the cancer cell's ability to survive the ADC's damage.
That's brilliant.
And the study showed it's especially effective in tumors that still have an intact TP53 gene. Because once you remove that BCL-XL blockade, TP53 just steps right in and finishes terminating the cell.
It just goes to show we don't always need to invent entirely new, crazy toxins. Sometimes we just need to disable the cancer's internal defenses so our current drugs can actually work.
Exactly.
Which honestly brings us full circle to the question of existing drugs. You know, we spent so much time on this show talking about million-cell atlases and bivalent radioligands and gating logic.
Yeah, the highly technical stuff.
Right. But sometimes a profound breakthrough comes from a generic bottle sitting right in your medicine cabinet.
I love this study.
Me too.
I know.
Let's talk about the Weizmann Institute study. They found that sildenafil, which most of the world knows as Viagra, could potentially reduce prostate cancer metastasis.
Yeah, a huge finding.
Now, my immediate obvious thought was blood flow. because that's what the drug is famous for. But the researchers made it crystal clear this has absolutely nothing to do with blood flow.
Nothing at all. It is entirely about internal cellular plumbing.
Okay, break that down for me.
So we have to trace a specific cascade here. Sildenafil is part of a class of drugs called PDE5 inhibitors. By blocking that PDE5 enzyme, the drug causes a major increase in levels of a molecule called cGMP.
Okay, hold on, stop right there for a second. So you block PDE5 and cGMP builds up. Why on earth does a cancer cell care about cGMP?
Because that buildup of cGMP acts as a massive roadblock for something called NPC1.
And what does NPC1 do?
It's a protein responsible for cholesterol trafficking inside the cell. So when cGMP builds up, it completely disrupts that NPC1 transport system. The internal movement of cholesterol just grinds to a halt.
Wait, why does a prostate cancer cell need to move cholesterol around internally in the first place? I usually just think of cholesterol in terms of heart disease, not oncology.
Well, cholesterol is a fundamental building block for cell membranes. It dictates how rigid or flexible a cell is. And when a cancer cell is preparing to metastasize, it has to undergo immense physical changes. It has to alter its shape, break its physical bonds with the main tumor, and squeeze into the bloodstream.
That sounds physically exhausting for a cell.
It is. And then it has to survive the sheer fluid stress of traveling through the body and eventually anchor itself into a brand new organ.
Wow.
To execute those gymnastics, its membrane has to be incredibly dynamic and flexible. And to keep that flexibility, it requires a constant, highly organized supply of cholesterol.
Oh, wow. So if the sildenafil cascade disrupts that internal cholesterol transport, the cell basically loses its structural integrity.
Exactly.
It loses the physical flexibility it needs to pack its bags and travel through the bloodstream.
Right. It becomes rigid. It becomes fragile. It essentially just gets grounded.
That is mind-blowing.
And the data backing this up is exceptionally robust. The Weizmann researchers didn't just look at a few petri dishes. This spans mouse models, human cell cultures, and a massive 20-year database from Cloud Health Services.
That database finding was the part that really fascinated me, because it showed better survival rates for patients taking sildenafil, but specifically, and this seems like the crucial detail, when they took it alongside cholesterol-lowering statins.
Yes, which perfectly validates the biological mechanism we were just talking about. It's basically a two-front war on the cancer supply chain.
Right.
The statins systematically lower the overall production of cholesterol in the patient's body, which starves the tumor of raw materials.
And then the sildenafil comes in and disrupts whatever limited cholesterol the cancer actually managed to scrounge up.
Exactly.
So the statin cuts the external supply line, and the sildenafil destroys the internal distribution network. That is amazing.
It's a really powerful combination.
But, I have to play the role of the grounded patient here for a second.
Of course.
Because someone is listening to this right now, looking at their medicine cabinet and wondering, wait, does this mean I should be calling my doctor for a sildenafil and statin prescription this afternoon? What is the actual takeaway for a patient today?
It is incredibly important that we are very clear on this. Do not rush out for a prescription based on this study alone.
Right.
The strongest human evidence presented here is observational. And observational data proves an association. It does not prove causation.
Meaning, we know the patients taking these drugs lived longer, but we can't mathematically prove it was the drugs themselves that caused it. It could be some other shared lifestyle factor.
Exactly. Without a controlled, randomized environment, there are always confounding variables.
All makes sense.
But what this study absolutely proves, beyond a shadow of a doubt, is that oncology must look at a patient's entire metabolic state, not just their genetics. The structural mechanics of a cancer cell rely heavily on systemic metabolic factors like cholesterol.
But rigorous randomized clinical trials are absolutely required before this combination becomes a standard of care.
100%, yes.
Right. So it's a flashing neon sign pointing us down this brilliant new research path, but we aren't at the final destination quite yet. We still need the trials. But stepping back, it completely changes how you look at the ecosystem of a patient's health. We aren't just fighting rogue DNA anymore. We're fighting the supply chains that feed it.
We really are. And, if I can leave you with a final thought to ponder today, I'd really love to build on the implications of that 20-year database discovery.
Please do.
What if the future of oncology isn't solely reliant on synthesizing new, highly complex, astronomically expensive compounds.
Right.
What if the next great leap in survival rates comes from using artificial intelligence to run complex pattern recognition algorithms over millions of existing health records?
Wow.
We might just find that the perfect synergistic combinations of cheap, safe, existing medications, drugs we already have sitting on pharmacy shelves right now are holding the secret to freezing cancer in its tracks.
That is an incredibly powerful thought to end on. The answers might literally already be out there, just waiting for the right algorithm to connect the dots. Thank you all for joining us on this deep dive into the data. Stay strong and fight on.